Now it's time to move towards our next presentation, which is from Dr. Felix Scholkmann. I hope I pronounce the name correctly. Felix is from Biomedical Optics Research Laboratory from University of Zurich, Switzerland. And the title of Felix's presentation is Changes in Water Properties in Human Tissue After Double Filtration Plasmapheresis, a case study. This paper looks very interesting to me, so I will send the link to the paper. So please, Lawrence, could you please start the video?
Thank you for inviting me to this webinar. My name is Felix Scholkmann. I'm from the University of Zurich in Switzerland, and I'm going to present my research results on plasmapheresis studied with Aquaphotomics. Our human blood is a mixture of about 45% blood cells and 55% plasma. The plasma is composed mainly of water, about 92%, but also of proteins and other solutes. Some of the substances in a blood plasma can be quite problematic or even toxic to the human body, like environmental toxins, damaged proteins and lipids, or autoantibodies. Autoantibodies are a group of antibodies, that means immune proteins that mistakenly target and damage specific tissues or organs of the body. That begs the question, is there a method that can selectively remove these unwanted substances from the blood? Such a method exists. It's called plasmapheresis, basically blood washing. There are two types of them. The first type is single filtration plasmapheresis, quite often used in the clinic. In this technique, the blood is continuously purified by passing the blood extracorporally through a filter and separating it into its two components, the blood cells and the plasma. The plasma is then removed and replaced with fresh donor plasma. The blood cells and fresh plasma are then returned to the body. This is done continuously until all the blood in the body has been purified. However, the method has some disadvantages. Firstly, it requires fresh donor plasma, and secondly, importantly, endogenous substances are removed with the filtered plasma. A further development is double-filtration plasmapheresis. Here the body's own plasma is not completely removed and discarded, but only the problematic components are selectively filtered out. The process runs with two filter processes in succession. The dirt filtered out, that means the problematic plasma components, is called eluate. This method was developed by Japanese researchers in Tokyo, and the first clinical results were published in 1980. The photo shows a patient who has had a double filtration plasmapheresis performed. In the paper, case studies of 7 patients were used to show that the method could be successfully applied in therapy for various diseases, such as in the case of atherosclerotic circulation failure, chronic renal failure, progressive systemic sclerosis, and rheumatoid arthritis. It was also shown that the technique significantly improved the blood flow to the legs. The method, therefore, has a positive influence on blood circulation. Here are shown different devices with which plasmapheresis can be done. The two devices on the right are able to do a double filtration plasmapheresis. The device on the far right was used in the study presented here. the INUSpheresis device. On the left is a typical eluate. Note the rather dark coloring compared to a typical blood plasma. Double-filtration plasmapheresis has been successfully used for the treatment of many diseases. Here's an overview based on published studies. Successful treatments were documented, for example, for Guillain-Barré syndrome, rheumatoid arthritis, inflammatory polyneuropathy, multiple sclerosis, acquired thrombotic thrombocytopenic purpura, COVID-19 and long COVID, and cancer. Positive effects have also been documented for the treatment of Alzheimer's disease. In this case, for example, a reduction of the chemokine RANTES by about 65% and a reduction of tumor necrosis factor alpha by about 18% could be demonstrated. What interested me and what I investigated in the present study was the question: is there a change in tissue hydration and water properties after a double filtration plasmapheresis treatment? The experimental protocol was as follows. Plasmapheresis was performed on myself twice. The protocol lasted 5 days. On each day I performed spectroscopic measurements on my hand. On day 2 and 4, plasmapheresis was performed, and on these days I also examined the eluates spectroscopically. For the spectroscopic measurements I used a micronear 1700 ES ultra-compact near-infrared spectrometer that enables to measure in the wavelength range of 908 to 1676 nanometers with a resolution of 6.2 nanometer. An integration time of 11.7 milliseconds was used and an average of 1000 scans employed for each spectrum to ensure an optimal signal-to-noise ratio. 15 consecutive measurements were performed for each measurement. Shown here are the spectra I obtained by measuring my hand on all 5 days. You can clearly see that the measured spectra correspond primarily to the water spectrum. The measurements were all taken at the same temperature of the hand, 31.5 degrees Celsius. To detect the fine differences in the spectra, the difference spectra were calculated. Here, the difference of the spectra before and after plasmapheresis was calculated. Here you can see the different spectra for the case after the first and second plasmapheresis. Clear maxima and minima are visible. For further analysis, the spectral information was analyzed using the 12 Aquaphotomics water matrix absorbance coordinates. The eluate was also examined spectroscopically. Here you can see the measured spectra. And here are the difference spectra. Here's the presentation of the results for the hand measurements as an aquagram. The red-orange lines are the spectra after the first plasmapheresis, the green after the second. You can see that both spectral pairs show a similar spectral pattern. The following was found: clear differences in the spectral features in the first O2 spectral region of water. The strongest increase was in C6, corresponding to the water hydration band, the HOH bending mode, as well as the OH stretch vibration mode, linked to the hydrogen bound strength of the water molecules. The strongest decrease was in C4, corresponding to water hydration shells, hydrated superoxide water clusters, and the H2O symmetrical stretch vibration. In general, there was a decrease in C2 to C4 linked to a weaker hydrogen-bounded water, an increase in C5 to C11 linked to free water molecules and small water clusters. The following can be concluded. These specific changes in spectral properties were evident after the first as well as the second double-filtration plasmapheresis treatment, whereas the first treatment caused the most pronounced effect. After the first plasmapheresis treatment, the number of small and large water clusters increased, whereas after the second plasmapheresis treatment, only the number of small water clusters increased, while the larger ones decreased slightly. In the case of the analysis of the eluate, the following aquagram was obtained. The strongest decrease in absorbance was at C6, associated with the water hydration band, as well as the HOH bending mode and OH stretch vibration mode. Fascinatingly, the specific water absorbance spectral pattern of the eluate is thus complementary to the measured in the tissue. This figure shows the aquagrams for both measurements in parallel, the measurement on the hand and the measurement on the eluate. What I also did was to have my eluates examined to see what toxins they contained. The results show that my blood was quite loaded with toxins. The 10 highest detected concentrations were aflatoxin, chromium, lead, cadmium, arsenic, lindane, cobalt, polycyclic aromatic hydrocarbons, disulfotone, and aluminium. It is also clearly visible that after the second plasmapheresis, the concentrations were reduced significantly. In addition, the presence of nano and microparticles was investigated in the eluate. Such particles were indeed found. The figure shows examples of particles containing titanium, aluminium, and silicon. The publication on this is currently under review. In summary, the following can be concluded about our spectroscopic investigations. In our study, we have shown that the water properties in the tissue change after double-filtration plasmapheresis treatment with the INUSpheresis technique with a TKM58 filter. Double-filtration plasmapheresis caused an increase in free water molecules, small water clusters, and a decrease in hydroxylated water clusters, superoxides of water solvation shells, and a weaker H-bounded water. The opposite effect was observed in the eluate of both DFPP treatments. The changes in tissue water demonstrated by our case study suggest that the positive physiological effects of the double-filtration plasmapheresis in general and of INUSpheresis with the TKM58 filter in particular may be associated with improvements in water quality in blood and tissues related to the respective water molecular structures. Such an improvement in water quality could, for example, be associated later on with improved blood circulation and optimized metabolic processes. Thank you for your attention.
Thank you very much, Felix, for this nice presentation and your work. So now the floor is open for questions and discussion. I'm not sure whether Felix is present today to answer the question.
He was supposed to be here, so I'm surprised that he's not. And what a pity.
Yeah.
I'll try to answer some questions if there are.
Yeah, there is one question from Medal. Medal, would you please like to speak? Would you like to ask your question?
Can
Or
you?
otherwise,
Pierre.
Hi, good morning, everybody. Can you hear me?
yes, we can hear you.
Okay, well, can you see me? No, obviously not. But anyway,
Not
doesn't
yet.
matter. Not
No.
yet. So I muted the camera. I tried to unmute it, but it doesn't work. Well, for whatever reason. Thanks for this great meeting here. This is really fascinating stuff.
Yeah.
I probably will have to ask Felix directly because I have a direct thread to him anyway. But I wonder whether the process itself, the DFPP process itself, could release secondary, let's say, contaminants from the filters that he used back into the bloodstream. Could he exclude that some of these debris are reintroduced in this process into his body? And secondly, I mean, how biocompatible is this plastic material in the case of such a, I would say, holistic approach? Because the entire body is affected. And I'm not quite sure in the manufacturing process of these devices, what kind of precautionary principles the manufacturers take care of to exclude that any of these, let's say, synthetic materials, either solid or, let's say, in volatile fraction, can be introduced into the organism when exposed to it. So this would be my question. Could you hear my questions?
Yes.
Yes. Here, I think biocompatibility is the number one question that has to be answered in such devices. I cannot comment on this, but what I've been thinking about this work was the filtering of the filtration of the blood. Filtering water changes the water structure, filtering itself. So this is something that probably deserves more attention and more work in the future. How filters themselves change. What is the contribution of filtering to the blood, water structure in the blood, and how it influences the whole organism? This is something that requires further attention, I think.
Romana, may I add another, let's say, question to that? I mean,
Yep.
I was following his approach, his kind of self-test approach when it started all the way back 2.5 years ago. So we have had several occasions to talk about it. And one issue is that in this process, he needs to do this quite regularly, because otherwise he ends up getting rid of this stuff, this toxic material. But as he's exposed and continuously to the environment he lives in, the food he eats, the water he drinks, it's a kind of accumulating back to original levels. So how can, I mean, this is a nice approach for a short-term treatment, but you can't do this, let's say, on an annual basis. It's quite expensive. Such a treatment is 5,000 euros.
I agree, but there are elements that you don't acquire for 1 year. There, I mean, there.
Yeah, he did not eat all that aluminium in 2 days.
Yes. So I mean, everything related to Alzheimer and high cholesterol and all these things are not the result of living 1 year.